started, the best way for you guys to know what you need to know is for me to show you old tests because material hasn't changed. So I have an old test here for you. I know. Alright, so final practical. This is usually not too bad. Okay, so first station would be something like this. What enzyme is used to convert hydrogen peroxide into less toxic compounds?
Catalase.
That is catalase. So you got it. Okay. What are the two compounds that you convert hydrogen peroxide into? Water and oxygen gas. Please don't miss that. That is easy. And yet, one of the most missed questions.
You're posting these, correct?
No, I am not. Why would I post these? This is a test. Okay, so in this, what's going on in this tube? You guys remember what this tube is? This is the citrate tube. So, which of these two tubes is positive?
Blue, the B one.
The blue one. The blue one's positive. You guys need to look over that sheet or the PowerPoint that I gave you because this is all over it. And if you miss that, that's just too bad. I will be disappointed in you because this is right in the middle of it and you guys need to know what it means. So, if you're looking for citrate metabolism, that means that you can use citrate as a carbon source. What is the indicator? Bromomethyl blue. Okay, that's really easy. Okay?
I can't read this.
Alright, you guys remember the antibiotic discs? What was this called?
Kirby-Bauer.
This is the Kirby-Bauer test. So, which antibiotic disc is best? A, absolutely. Now, what is the area around that disc called?
The zone of inhibition.
How about an area of inhibition? Y'all remember that? Now, what do you think is going on in C? Antibiotic resistance. So how do you get antibiotic resistance? What are you doing to the antibiotic? In general, the way that they deal with antibiotic resistance is by destroying the antibiotic. So they can grow right on top of the disc because they've destroyed the antibiotic itself. Most of the time they have a enzyme that breaks down the beta-lactam ring in antibiotics. Okay, remember that's that square unit that's in most antibiotics. They're able to break the ring, and that's how they do it. All right, the experiment we just went over today, you either have bacteria growing or you don't. Okay? What is the purpose of ampicillin being added to that plate? Well, what happened on the place that didn't have ampicillin? Everything grew, so you can't tell the difference one way or the other. The ampicillin allows for you to differentiate whether or not the plasmid was taken up or not. With me? So it kills all the cells where you didn't have the plasmid taken up. You guys had some blank plates because you didn't take up a plasmid and it killed everything, right? So that's what the ampicillin is for. What is the term by where you take up DNA? What was the term I called this? Transformation is a good one. Okay? You're transforming the bacteria. You're giving it DNA it didn't have before. Now, I guess you guys can't see that, but there's a lot of colonies on this plate. sometimes you get green and white colonies. Can you guys guess to why you get both green and white colonies? Not so much? Well, on the P-glow you have antibiotic resistance, right? We just went over how antibiotic resistance works. So how does antibiotic resistance work? You destroy the antibiotic. So if these green cells are destroying the antibiotic, then the other cells are able to grow too. Those are called satellite colonies because the transfected cells are destroying the antibiotic. Does that make sense? Okay? Alright, so we didn't do the nitrate test, so you don't have to worry about it. Now, y'all remember we went over this specific thing. It is in your PowerPoint. Now, we did this in lab in that like multi-tube, right? However, I want you to know how to read these kind of tubes. It is important to me. It will be important to you. So, what is this result? It is black. It has red at the top, which means what? Remember how you read these tubes? Slant over butt. And you're looking for fermentation, right? So what you got is no fermentation over fermentation. So that would be K over A, right? So K over A, and then you go gas production and sulfide production. Yeah. Is there any gas?
Yeah.
No, so negative. Is there any sulfide? A whole lot of it. So it'll be negative and positive. So K over A, negative, positive.
So K is fermentation.
K is no fermentation. A is fermentation. But when you get black, you have to assume A. And I know the black is going up here, but you have to look at the very top, and that's red, so that's no fermentation. Because these tubes start out red. Yellow is fermentation. You guys with me? All right, next tube, what do we got? Color over color.
K over K.
Well, it's fermentation over fermentation, right? So that would be A over A.
Oh.
Okay? Did we get any black precipitate?
No.
But did we get gas?
Yes.
Okay. Now, in normal circumstances, I would have you guys do the lettering scheme. Do K over K, gas, or the plus and minuses. Actually, since we did not do that in lab here, all I want you to do is be able to read this. So, for me, if we had a test question like this, if you told me fermentation over fermentation, and then you said gas production, that would be right. Okay? I am making it easy for you. Okay? You do not need to know the lettering scheme. You just need to tell me slant over butt, fermentation over fermentation, and what's produced. And it's sulfur reduction. Tell me sulfur reduction. I'll take black precipitate. Gas is produced. Tell me gas is produced. Okay? Any questions about that? Just tell me what you see, and that's a viable answer. Okay, but you do need to be able to see them. Remember what kind of sugars are present. Now, I told you the specific types on the PowerPoint. However, I would be willing to take the slant has disaccharides, so 2-part sugars, and the butt is just a 1 sugar, glucose, just a single sugar. Okay? So disaccharides over single sugar. Okay? Does that make sense? So if it just has the ability to deal with, or just has the ability to ferment the bottom, what does that tell you? It can't ferment disaccharides. Okay? It can only do single sugars. Make sense? This is the level of understanding I want you to understand, or want you to get to. If it can't do the top, and the top has disaccharides, but it can be the bottom, which is single sugars, and you say it can ferment single sugars, but it can't ferment disaccharides. So what's the difference between the two? Well, you have a bond in between two sugars. You might not have that enzyme. Make sense? All right. In general, that's as much as I want you to get into on that one. Yes,
Confused on the, so it's just always, the butt is always disaccharides?
No, the
Or
butt-
polysaccharides?
the butt is always glucose. It's always a single sugar.
Like
Every single time.
all four of those have glucose at the bottom?
Absolutely.
And then all four have disaccharides at the top?
Yes. Okay, they're made in parts actually. So you make the butt first, and then you make the slant after.
So even though it's asking about sous vide, it'd be the same for, in this situation, but it all have the same sugar
Every
content?
one of them has
Okay.
the same stuff in these tubes.
Okay.
So what you can make a distinction is if a bacteria grows on any of those tubes on just the butt, it can only live on monomeric sugar or glucose. If it is growing in everything, it must have the enzymes to use disaccharides.
Okay.
Okay? No problem. You guys got to remember coagulase, okay? We did this. Why would an organism produce coagulase? They don't want to die. They want to form a clot. It can be a whole bunch of different answers. That's open-ended. Okay, which one's positive?
The one with the quads on it.
A, absolutely. Okay, y'all remember this? What organism produces urease? Hot pink. Okay, remember that is positive. Why is it positive? What is urease? Urease is an enzyme that breaks down what? Urea. So what's happening to the pH of that tube?
So basic.
It's getting more basic. That's true. This is what I want you guys to think through, because just because the questions on here are these kind of questions, these are not going to be the questions on your test. Listen to the words that are coming out of my mouth, because those could be the kind of questions that are going to be on your test. Okay? All right. What's the differential agent within that tube? What are you looking for? You're looking for
Urease.
what enzyme?
Urea.
Urease.
Urea.
Okay, so if that's the enzyme you're looking for, well, what's in the tube?
Erase.
Urea. Okay, urease breaks down urea. Okay, so the tube has urea in it. Where does the urease come from? It's the bacteria. So you don't have to add it. It comes with the bacteria. It's packing a lunch with urease too. Okay, you don't have to add that. So you're looking for the ability of bacteria to produce urease or not. Okay, so the tubes have urea. That's the differential agent. That's all they got. You add bacteria, and the bacteria produce that enzyme that can break down urea. You also need to know the pH like we talked about. Please do not hang up on that. Okay, methyl red test. We haven't done one of these, but we talked about it. Remember, there's methyl red and Volks-Proskauer. Y'all remember what Volks-Proskauer does? You need to know Volks-Proskauer. Okay?
That's the rust colored.
What's that?
Is that the rust?
That is the rust-colored one, but the rust-colored one is looking for what kind of byproducts of fermentation?
Acid.
No. What is the other one? It's either acidic byproducts or neutral byproducts. Volks-Proskauer tests for neutral byproducts of fermentation. That means the bacteria don't like acid, so they make acid in fermentation and then they destroy it. Okay? Remember that? Neutral byproducts is Volks-Proskauer. Methyl red tests for acidic byproducts. So if you add methyl red and it stays red, positive for acidic byproducts. With me? Usually the pH for this is around 3. Okay? We just talked about this. If you have bacteria and they're surviving ultraviolet light, what are they doing? They produce a pigment. What does UV light do to DNA? Causes DNA damage or double-stranded break. Actually, this is cysteine dimers. Thymine dimers. Okay? All you need to know is DNA damage. That's enough. Okay? Thymine dimers is what happens. You do not have to know this. What I want you to know is bacteria can produce pigments to absorb ultraviolet light. That's good enough. That is more accurate. Okay, this is a picture of blood auger. How do you feel about that picture of blood auger? What kind of blood auger is this? You guys can see straight through and read through it. What does that mean? What kind of hemolysis is that? Beta is when it's complete hemolysis. So you can see through it. That's beta hemolysis. So, beta hemolysis is what we're seeing there. What kind of organism can do beta hemolysis? You should know too.
Streptococcus.
Streptococcus pyogenes is a great example. What's another? What's another bacteria that causes impetigo?
Staph aureus.
Staph aureus, Staphylococcus aureus, or Streptococcus pyogenes. These are great examples. All right. You definitely have to know what the indole test is for and what it does. What's the positive?
Red on top.
Red on top. Okay. What amino acid is being broke down in this test? What's the amino acid in turkey?
Tryptophan.
Tryptophan. Okay, you're looking for tryptophan being broken down, and that's where you get the indole positive from. Okay? So you're looking for the enzyme tryptophanase. That is the enzyme that breaks down tryptophan. And if you know tryptophan, the enzyme that breaks it down should always be tryptophanase. All you have to do is add an ACE on something. That's the enzyme that kills it. Okay, remember this test? That is the motility media. We call that the SIM tube. So, this is the SIM tube, and it's testing for the ability of bacteria to either stay where you put them or to move all over. And that is a motility test. So you're looking for the presence of flagella or motility.